All right, now we're going over microbial growth. Okay, part of it is how we grow microbes in the lab, but part of it is understanding what makes bacteria grow and what makes other microbes grow. If we can understand how to do it, we can understand how to stop it. Okay, so without further ado, let's talk about it. Okay, so the requirements for growth. Okay, the physical requirements are there, the chemical requirements are there. Okay, so let's explain that in a little bit more detail. Okay, so first of all, temperature. Okay, there is an optimal temperature at which certain bacteria function. Depends on which bacteria. Okay, if you're a bacteria that lives in a human body, then your optimal growth temperature will most likely be exactly at or right about human body temperature. Okay, it's slightly different for those that live in the nose and stuff. Those need it slightly cooler because they live in slightly cooler temperatures. But if you're infecting a human, it's best to infect it at the temperature it grows at. Okay, now there is a minimum temperature that it functions at and a maximum temperature it functions at. Okay. All right, so let's look at these three. Okay, cycrophiles. Okay, really function well at very cold temperatures. Okay, they function very near freezing. Okay, remember in Celsius they freeze at 0. Okay, cycrotrophs. Okay, live more closer to normal, like cold temperatures, but not like refrigerator temperatures or freezer temperatures. Okay. Mesophiles. Okay, meso is middle. Mesophiles love the middle temperature, which is about 37 degrees and stuff. So right here, that's us. Okay. Thermophiles live in very hot areas, 60 degrees and stuff. You'll find that in soil, especially in compost piles and stuff. They warm up a lot. Okay. And then you see the hyperthermophiles, which exist in like hot springs and underwater sea vents where there's literally boiling water shooting out of it, and therefore, like, you can literally have water above 100 degrees. Okay. Okay. So cycrotrophs live between 20 and 30, between 0 and 20 and 30 degrees. These are the ones that usually cause food spoilage, because again, they're living in your fridge and they're living at room temperature, not body temperature. Okay. Okay. So first of all, when we talk about stuff, I'm talking in the Celsius scale, not the Fahrenheit scale. Okay. But this is the dangerous temperature, right? The rapid growth of bacteria is between room temperature and kind of high, but not cooking temperature. Okay. All right. When you freeze stuff, right? You really don't have a lot of growth of bacteria. The colder you get, so your freezer is -20 right here. There's very little growth right there. Okay. Okay. By the way, this is why you don't put a lot of hot food in your freezer all at once, or fridge all at once. Okay. If you put in a lot of hot food, you warm it up to a significant amount of time where the food is in this warm range. Okay. See how fast this comes out of the warm range and how long this stays in the warm range. So Okay, most bacteria grow between pH 6.5 and 7.5. Okay, molds and yeasts are actually slightly better at growing in acidic environments, but between 5 and 6. Acetophiles grow in acidic environments, and we'll talk about like those later. Okay, especially the bacteria that grow in your stomach and stuff. Okay, hypertonic environments. Higher tolerance inside the cell cause plasmolysis due to osmotic pressure. Okay, and this is actually a really important concept. So if you add salt, this is what happens to bacteria, and bacteria usually dies when this happens. Okay, even if it doesn't die, it doesn't reproduce. It just kind of stays there. This is really important. This affects us in many ways. Okay, first of all, before refrigeration was popular, most people salted their food. Okay, you take fish out of the water, you literally add a whole crap load of salt to it. Salted fish. Okay, when bacteria encounter salted fish, if they land on the fish, this is what happens to them when they die. Okay, when you go home with a salted fish, it's literally as hard as a rock. You just wash out the salt, and once you wash out the salt, you can eat it. Okay. The other thing to do that we still use this for is jams and jellies. Okay. The stuff, the osmotic pressure doesn't have to be caused by salt. It can be caused by sugar. Like jams and jellies have really high concentration of salt. Okay, and that really high concentration of sugar, that really high concentration of sugar also prevents bacteria from growing. Okay, in fact, one of my first lab jobs, I had to cut slices of brain, and the brain was preserved not in some of a preservative, but in 50% sugar solution. And because that was so high sugar that the bacteria couldn't eat it. And so you could just take that sugar solution and keep the brains in there for a very, very long period of time without any damage happening to them. And if you needed them, you just wash out the sugar. Okay. 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 So make sure you understand these guys. Okay. Okay. Blah, blah. Okay, carbon is the structural backbone of organic molecules, right? We use carbon to build things. Autotrophs can use CO2. Other heterotrophs can use things that are derived from autotrophs using CO2. Okay, but we all need carbon. Okay, nitrogen is necessary for DNA, ATP, proteins. Okay. Most bacteria decompose protein material for nitrogen source. Okay, some bacteria can use NH4 plus or NOH3 minus from organic material that they consume. Okay, a few bacteria uses N2 air, the nitrogen in the air, for nitrogen fixation. This is really important because literally these bacteria are the only bacteria that can actually get nitrogen and break down nitrogen into NH4, which these guys can use, or amino acids, which these guys can use. So before modern technology, the only bacteria were actually capable of nitrogen fixation. Okay, and without that, we would have no nitrogen in our soil and stuff. Okay, so back in the old days, before commercially available chemical fertilizer, what people would do is they would grow soybeans every once in a while. They did something called crop rotation, where they grow whatever the crop they wanted, and then they grew a soybean crop, and then they grew whatever crop they wanted, and then they grew a soybean crop. The reason why that soybean crop was important was the bacteria that lived with the soybeans were very good at nitrogen fixation. Okay, so there was a lot of nitrogen being added to the soil while the soybeans were growing. That's why soybeans are actually a good source of protein, because the nitrogen bacteria put a lot of nitrogen into the plants, and the plants accumulate a lot of protein. Okay. Sulfur is used in amino acids, biotin, etc., etc. Okay. Phosphorus is used in DNA, RNA, ATP, found in phospholipid bilayer. Okay, and we recycle phosphorus. Okay, so phosphorus is very limiting. So without bacteria and funguses decomposing stuff, we will run out of phosphorus very soon. Okay. All right, so trace elements. Yep. Okay. Oxygen. Okay. Oxygen, vacuum, obligate anaerobes, facultative anaerobes, etc., etc., etc. Okay, so here, okay, make sure you understand this chart, because this chart will be on your exam somehow, somewhere. Okay, obligate aerobes. Okay, okay, these are obligate aerobes, which means they require oxygen. We'll skip here. This is obligate anaerobes. This cannot take any oxygen. Okay, so they only grow without oxygen. Okay, so now top of the vial, bottom of the vial. Okay, now that that's that, facultative anaerobes, they would prefer aerobic growth, but they can live without it. So there's going to be more of them near the top, but some of them on the bottom. Okay, because they grow better near the top, but they still grow on the bottom. Okay, aerotolerant anaerobes, they do not use oxygen, okay, at all, but they don't care that it's there. Okay, they literally just don't care. So they grow evenly throughout the vial. Okay, the last one is microaerophiles. Microaerophiles need a little bit of oxygen, very, very little, but they need it. So you can't go all the way down to the bottom because there's no oxygen there. You can't be at the very top because there's too much oxygen there. The oxygen levels have to be something like 1 or 0.1% of what it actually is, okay, in the air. So they can only live in the middle of the vial, okay. All right. So oxygen causes all this stuff, okay? And so this is why oxygen is slightly toxic. Our body has chemicals that can break down this stuff to water, okay? And this is superoxide. This is an enzyme called superoxide, this mutase that does this, and that protect us from the oxidative damage. When you don't have this at all, then you are an anaerobe. Okay. Okay. Okay, so you should be able to answer these guys. Okay. Okay. So more biofilm. So this is actual biofilm. Okay. Okay. So biofilms can be found in digestive system, sewage treatment plants. They can even clog pipes that have the slime growing on them. Okay. Okay. They're much, much more resistant to microbial sites because the slime actually prevents the microbial side from getting to the microbe. Okay. Okay. Different types of culture media. Okay. So some definitions. Okay. Cultural media are nutrients prepared for microbial growth. Okay. Sterile means by definition there's nothing living on it. Okay. Um, Inoculum is the growth of microbes into a medium. So I usually if you want to grow something, you take a small sample of it and inoculate your media with it. So that little sample you put into the media is called the inoculum. Okay, you inoculate with the inoculum. Okay, a culture is the microbes that are growing. Okay. Okay, agar is a complex polysaccharide. Okay. Okay. Um, yeah, I just want to make sure. Agar is a complex polysaccharide. Okay. And because it's such a complex polysaccharide, it is not metabolized by microbes. It comes from seaweed. So like bacteria that live on land has no reason to be able to break down seaweed. So we use agar with the expectation that we can't break agar down. Okay. Agar liquefies at 100 degrees. Okay, so you can take powdered agar, add it to water, boil it, okay, and it will melt. And once it melts, it will solidify about 40 degrees. So once it cools at room temperature, um, it solidifies. Okay. Uh, okay. Uh, okay. Sorry, I'm having a little malfunction. Okay. Okay. Uh, chemically defined media. Chemically defined media is not particularly useful. It is where the exact chemical composition is known. Okay. So 3 parts sugar, 1 part salt, 1 teaspoon of something. Um, exact chemical formula is known. Okay. A complex media consists of extracts of stuff, beef heart, um, stuff like that, like beef extract or yeast extract. Okay. Do we know exactly what's in it? No. Do we know roughly what's in it? Yes. Do we know the exact amounts of each chemical? No. Do we know approximately what it is? Yes. Okay. But it's complex. Okay. And so this is the stuff that we usually grow things in. So we usually grow things in either nutrient broth, which is like a soup like thing, or nutrient agar. So nutrient broth plus agar is nutrient agar. It's a thick, solid, yellowish thing. Okay? This is a chemical defined media. I know exactly what goes in it. Okay. Okay. This is not exact defined culture media. This is a complex media. Peptone is just partially digested protein. Beef extract, okay? Like you just basically boil the shit out of beef and to the point where it's like goo and that's extract. Okay. Okay. Reducing bacteria media, sorry. Reducing media. One of the things that anaerobic bacteria can't handle is oxidative damage. The reducing media deals with oxidative damage and it depletes the oxygen from the atmosphere. Okay. Um, okay. It reduces everything it touches, including oxygen, and therefore it depletes oxygen so anaerobic bacteria can grow on it. Okay. So here, you let this go. Okay. And this palladium will actually absorb all the oxygen. Okay. And we release carbon dioxide and stuff. Anaerobic chambers obviously can't be exposed. You can't expose it to air, right? Because that wouldn't work. Okay. Okay. Capnophiles are microbes that require high levels of CO2. Okay. So you either add a little CO2 packet. Okay. The CO2 packet is kind of like you can buy little packets of CO2 and just release them into the atmosphere. Again, this has to be sealed, right? So you seal in, you release the CO2, and that level goes up. Okay, or use a candle jar. The old school method is use a candle jar. So you put in your bacteria, you put in a little candle, you light the candle, you seal the jar. Eventually, the candle will burn out all the oxygen, and what's left, the byproduct of the burning of the oxygen is CO2. So you will have a jar that's filled with CO2. Okay. Okay. Biosafety level. Stuff. Okay. The definition of what biosafety is. Okay. So make sure read that. Okay. Um, if we were in lab, the lab would be biosafety 1. Okay. So nothing kind of. Okay, selective media. Selective media is designed to suppress unwanted microbes. And so you will have inhibitors to suppress growth of certain things. Like, for example, there are media that select for gram-negative cells and suppress gram-positive cells. And that is an example of a selective media. Okay, differential media has some distinguishing qualities of different microbes on the same plate. So here you can see blood agar. Okay, if the colonies can feed on blood, they will leave a little hole where the blood used to be. If it was a colony that could grow on the blood but not use it, it will grow on top of the blood, but it wouldn't use the blood. So there will be no hemolysis. Okay, so it doesn't matter that they can grow on it or not. It has to change the media somehow, or like here, the Staph aureus, changes it makes this more acidic, so it changes this area to yellow. Okay, so you can tell these two types of staph apart. Is one of them is yellow and one of them is not yellow. Okay. Okay. Enrichment culture encourages the growth of a desired microbe by increasing very small numbers of the desired organisms to detectable levels. So I can grab some bacteria and I can culture them all. If I found one that I want, I can selectively grow it in an enrichment medium. Okay. Okay. So the difference between selective and enrichment is that in enrichment, I'm actually trying purposely to grow as many of that bacteria as I can. The selective media, I'm just trying to see if it grows and see what grows. Okay. Okay. Uh, so describe pure cultures can be isolated by using straight. Okay. So by definition, a pure culture only has one species or strain of bacteria in it. A colony is um, comes from something called a CFU, colony forming unit. You drop a CFU onto a plate and it will grow into a colony. The way you do this is usually the streak plate method. Okay, so you pick up your bacteria and you smear all over versus number one here. Okay, when you first smear the bacteria, what you get is a very high concentration of bacteria. Okay, so here you see that eventually there's there's red dots and white dots, two different bacteria. But I can't tell because they're all mixed together. Okay, I smear this once, I then burn like heat my little spatula, and then I smear this number two. Okay, so now I am I'm barely touching number one. So I'm taking this little part of number one and smearing it. I'm taking this part of number one and smearing it. So that is going to be a much lower concentration, but still, it's still too concentrated. So I do it again. And so you can now tell at three that this isn't nearly as concentrated as before, but it's still too hard for me to pick a red dot from a white dot. So I'll do it again. And number four, now I can see there's very clear dots that are just red and very clear dots that are just white. And you can pick one of those up. And now I will just have the white dots. Okay. 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 Oops. All right. Okay. So deep freezing. Um, Between -50 and -95, this is where you're literally dropping into dry ice, not dry ice, but liquid nitrogen, and liquid nitrogen or a -80 freezer, like nothing happens to it. Okay, uh, lyophilization or freeze drying, you dehydrate it in a vacuum, and that becomes really dry and frozen, and you don't have to deal with that. Like they don't grow anymore. Okay. All right. Bacterial division. Uh, oh, you might not have heard that, but anyway. Okay, so binary fission I don't care about. Okay, what I do care about is generation time. Okay, generation time, by definition, is the time required for a cell to divide. Okay, it can be anywhere from 20 minutes, 24 hours. Okay, so if I start off with one cell and the generation time is 20 minutes, in 20 minutes, I have two cells. In 40 minutes, I have four cells. In 60 minutes, I have eight cells. Okay. The total number of cells, okay, is 2 to the n number of generations. And that's assuming you're starting off with one cell. Okay. Uh, if you're starting off with three cells or something like that, it is 3 times 2 to the n. Okay. So 1. So you can see that there. Okay. Okay. So you can see how fast they divide. Okay. After 20 generations, you get 1,000,000 cells. If they divide every 20 minutes or so, which is the maximum fastest division time, 20 generations is something like 3 generations, hour, 8 hours, less 7 hours, less than 7 hours. Okay, you can go from one cell to 1,000,000 cells. Okay, so it is hard to chart this in a normal scale. Okay, a numeric scale. So we use a log scale, and a log scale, exponential growth appears linear. Like, okay, so make sure you understand the phases of growth. So there is a lag phase. So once when you were growing bacteria, the first thing you do when you add bacteria to a growth media is a small lag phase. That lag phase is the bacteria adjusting to the new media, and there is very little growth at that time. Okay, the second phase is log phase. The log phase, the bacteria have adapted to the growth media and now they're growing exponentially. The log phase is where every generation time, the bacteria doubles. Okay. The next step is the stationary phase. Okay. As the bacteria doubles, they're using more and more resources. So they're competing more and more. At first, they're not really competing at all because there is gallons of this stuff. Okay, but at the stationary phase, not only are there much more bacteria than before, there's much more waste products than before, and there's less nutrients than before. So now the bacteria are kind of competing for what's left over. And at this stage, the number of living bacteria and the number of growing bacteria and the number of dying bacteria are equal, and therefore they don't grow anymore. Okay, and the last phase, the death phase, the two of them have caught up to each other. Okay, and now there's more dying than growing bacteria. You reach a death phase. The death phase does not mean all the bacteria are dead. Far from it. It just means more dying than actually growing. Okay. Okay. All right. So measuring bacterial growth. Okay, so let's talk about measuring bacterial growth. Plate count, filtration, most probable number count, right? Microscopic count. Okay. So plate counts are by far the most often of these, and so I'll explain that one. Skip the others. Okay. So plate counts. So you literally take a culture, you take a certain volume of bacteria, and you like 1 microliter, you spread it on a plate, and then you grow the plate, and then you count the number of bacteria. Okay, and so if I put in 1 microliter, microliter is 1 millionth of a liter, and I put it on a plate, and I get 10 colonies, that means there are 10 colonies in that 1 microliter, which means there are 10 million bacteria in that 1 liter solution I have. Okay, does that make sense? So I can backtrack to that. But here's a problem. The number of bacteria in a solution is going to be far higher than what I usually count like this. Okay, even if I put 1 microliter on there, I can easily get thousands, if not tens of thousands of colonies. And a human being can't really count more than 300, 400 dots on a piece of paper accurately without counting multiple dots or missing dots or just being flipping bored out of their freaking mind. And you literally have, when you're doing this, you're not doing this once just to learn how to do it. You're doing this all day. So to do counts accurately, you have to keep the count small. Okay? And so you can count reasonably quickly and efficiently. So what you have to do really is to dilute out the bacteria. Okay, so if I take the original inoculum, I separate this out. Okay, so 1 to 10, this is 1 to 100, this is 1,000, this is 1 to 10,000, this is 1 to 100,000. Okay, if I pour this out, that gives me 3 bacteria. That means this will be 30, this will be 300, this will be 3,000, and this will be 30,000. Okay, and so you can see like this is countable. This is easy countable. This is countable. This is borderline countable. This is impossible, and this is beyond imagination. Okay. All right. So don't worry about between the pour plate and spread plate method. Okay. Filtration. Okay. So there's the other method is filtration. So there are some bacteria that are so sparse that it's very hard to count. Like the number of bacteria in a droplet of water in a pond or something, or in a lake. Okay, that's hard to do. Okay, because most of the time you'll get nothing. So what do you need to do? You need to concentrate the bacteria. Okay, so what I need to do is take all the bacteria, pour it, take all the solution, take 1 liter of solution, pour it through a filter, and now all the bacteria is inside that filter. Take that filter, flip it into a petri dish, and count the colonies. And that'll tell you how many bacteria there are. Okay. So you can see that there. Okay. Okay. We'll skip most MPN, we'll skip direct microscope count. That's literally what it sounds like. You literally sit there and count shit. Did that before, sucks. Don't ever want to do it. Okay. Okay. There are other ways of estimating the number of bacteria methods. Okay. The first one is turbidity. bacteria are not clear. They're not optically clear. So the more bacteria that's growing in the shit, the more cloudy that shit gets. Okay. The measurement of cloudiness is turbidity. So the more turbid a solution is, the more bacteria there is growing in it. And this term only refers to microbiology. Okay. Turbidness in some fields has to do with the number of dirt and crap in the soil or in the water. But for microbiology lab, it has to deal with the number of bacteria. Okay. All right, so that ends the presentation. I hope you guys understood all that, and as usual, I'll be open for questions.